hello to everybody who is out there. Unfortunately, I can't see you. I hope you can see me. And it's a pleasure to be here talking with you today. It's events like the Gatsby which got me interested in doing science in general. And I hope that the experience is a positive one for you as well. So I'm going to talk to you today about work we're doing in the lab, looking at effectively how cells are organized in plants and how these configurations of cells give plant organs their functionality. So we have this very fancy sounding title, which I'm going to explain to you. The title being Topological Analysis of Multicellular Dynamics in Plants. And the explanation will be coming forward in the presentation itself. So what we're considering here is this notion of multicellularity, and this is a major evolutionary phase transition which has happened multiple times independently in both the plant and animal lineages. So over the course of evolution, multiple independent events have led to cells after they've divided, rather than continuing to exist as independent cells, of keeping them together. And this has led to the creation of multicellular complex life. So the reason these transitions have occurred, it is believed that these collections of cells, which are interacting with one another, are able to collaborate to achieve things that individual cells cannot. So if we look here, for example, in the bottom left, these are, I have got an example of animal cells here, but these are some cells which are growing in culture. I can't tell you what type of cells they are, other than they come from an animal. And they're kind of dividing and they crawl around a bit, and they don't really do very much. They do a little bit, but not very much, but they're largely existing as individuals. And then the question then becomes, how do you go from these sort of pseudo-independent collections of cells to something as complex and dynamic and incredible as an octopus? So the jump from this to this is really what we're trying to get at. And it is this notion of emergence that we're talking about here. So how do you have individual cells come together to achieve something above and beyond the sum of the parts? So how do we have an emergent behavior of, say, a collection of cells in culture versus a collection of cells which make up the octopus? And these are very much not the same thing. So towards this, we're considering multicellular complexity. So how do cells come together to create complex life? And now finally, I have some plant examples here on the bottom of different forms of simple forms and more complex forms of multicellular plant life. So in the middle here, we have these filaments, and then we have this volvox, and on the right here is a cross section where you see all sorts of different cells and cell types and different arrangements. And the question what we're trying to get at here is, how do the cells come together to create complex life? And another way of addressing this is to ask, what is the relationship between how cells are organized and the function of the organs which they come together to create? And in a more simple way of asking the same question, why are cells organized in the way that they are? So we're looking at plant organs as integrated systems of cells. So the idea here is that the cells which make up plants, they're glued together by shared cell walls, so the cells don't migrate. So in the case of our body, or in animals, the cells have the capacity to rearrange themselves. They can move. And in plants, they cannot, and that's because the cells have cell walls and they're stuck together. And what that means is these organs, which are assemblies, complex assemblies of cells, are spatially constrained three-dimensional cellular networks. So they're spatially constrained because they can't undergo rearrangements. It's not unlike a network of roads in a city where you're constrained by space and the roads largely cannot move. So in that sense, you have this constraint of space in the creation of a plant organ, which you would not have in the case of an animal organ. And these cell-to-cell interactions are important in the context of both transport and communication. So if we look at these patterns of these phylotactic patterns that we see in these different plant species at the bottom, The mechanism by which this patterning is occurring is through cell-to-cell communication. So the construction of these complex arrangements of cells is mediated through the self-organizing and patterning process of cell-to-cell communication. And then ultimately, once the organ has formed, the functionality of the organ is maintained through cell-to-cell communication. So what that means is there's a very important relationship between how the cells are organized and how the organ is functioning, given that there is a relationship between the structure and the function at the macro scale in terms of these phylotactic patternings. But we're also going to be looking now at the cellular scale within the organs as to what the relationship between form and function is. So to try to address this, we developed a method to quantify the patterning of cells in plant organs. And I'll take you through this briefly now. We begin with a plant tissue, and the tissue is stained with a fluorescent stain which labels the cell wall. And the cell walls, of course, are just exterior to the cells themselves. So if we have a cell wall stained fluorescently, what we effectively have is like a skeleton marking the boundaries of where the cells are located and their shapes. So by staining the cell wall, we can use this boundary marker to perform a process called segmentation. And what the segmentation is doing is it's identifying the unique cells within the tissue. So up in the top left here, we have, this is an Arabidopsis hypocotyl, which we'll talk about more shortly. So in green, what we can see is the fluorescently stained cell wall. And then we perform the segmentation process where we identify the individual cells. And that's not the actual color of the cells. The different cells are given different colors to indicate that they're unique segments. So using this computer algorithm called watershed, we can identify the 3D shape of each of the cells by using this fluorescent cell wall marker as a boundary. And then after that, we can cast what's called a mesh, a polygonal mesh, over the surfaces of the cells. And from that, we can identify, we can discretize the 3D geometry of each individual cell that we have segmented. So this is quite powerful in the sense that we can digitally capture in 3D the cells which are making up the tissues. Then the next step that we use in this approach is to then look at which cells are physically connected to which other cells using this mesh, which is describing the surface. And from that, what we can do is extract a network which tells us which cells are connected to which other cells. So here the network, the nodes, which are these dots, are representing the cells themselves. And the lines which are connecting the nodes, called the edges, these are the physical associations between the cells. So what we've done is gone from a 3D picture of a plant tissue down to a network which describes how the cells in the plant tissue are connected to one another. So once we have that network, we can then begin to do a topological analysis or a network-based analysis to understand the properties of the arrangements of these cells. And I will take you through a couple points on this. So what's the key point in this regard is that we're able to discretize the organization of cells. So discretize in the sense that we can take a picture and turn the picture into numbers. So we can then take what is effectively a qualitative piece of data in the picture that we have and distill it down into something which is quantitative, which is this network describing how the cells are connected. So then through the topological or network-based analysis of these cell connectivity networks, which I'll describe on the next slide, we can quantify patterns and properties of patterns using network science. So we can actually start to attach numbers to how the cells are organized within the tissue. And what these networks are representing are roadmaps of potential communication between cells. So in the plant organ, as I mentioned, the cells cannot move. So cells can only communicate with their immediate neighbors and their neighbors of neighbors through the intermediates. So what we are building up effectively is a map describing the potential routes of communication through the plant organ. And a final point is in these images, because we're using confocal microscopy to obtain these 3D images of cell wall stained tissue, we have managed to capture the whole organ and all of the cells So what it means is when we do this image analysis, we have all of the cells represented within the organ, and there's no data that are missing. And the quality of the data, the confidence with which we're able to establish these connections is very high because we're doing a physical-based image analysis. And this is a departure from other types of biological networks. For example, in the case of looking at protein-protein interaction network data, there are different techniques which each can give indications as to which proteins are physically interacting, but the overlap between them is not always one-to-one. But this is a different type of data, so I don't want to be saying bad things about protein-protein interaction data. Just to say that we're operating here at a different scale. Rather than looking at the interactions between proteins, we're looking at several scales higher and looking at the interactions between the cells in tissues. So in terms of abstraction, we can go from these images here on the left, and this is, again, the hypocotyl, and what we're looking at in white, these are the individual cells, and in yellow, this is the mesh that's been cast on top of the cells. And we can do the image analysis to see where the mesh in one cell is connected to a mesh in the next cell, and then turn this into a network. So we go from these very large TIFF image stacks, which are around 1.8 gigabytes in size, and we can condense it into a text file, which is 160 KB. This is just one example. But it's a 10,000-fold abstraction in terms of data size. So we go from a very large file to a very small file. We go from effectively a qualitative data set to a quantitative data set, which we can now start to analyze. And just to show you some video bling, because everybody likes to see a rotating 3D something or other, this is the Arabidopsis hypocotyl, and the balls are the cells, and the green lines are the physical connections between the cells. And the different cell types have been identified and given different colors. So you can see in the middle you have the endodermis, which is in purple, and you have in, I guess that's cyan, the second outer layer, that's the cortex. Just to give you an idea, a visual idea of what the underlying network data look like if you were to lay it out in 3D. But this is strictly the network. This isn't actually the geometry of the organ. This is just the connectivity of the cells. Okay, so I can't take credit for this idea of mapping the connectivity between cells, but this man here can. He's possibly Spain's greatest scientist. This is Ramon y Cajal. He won the Nobel Prize, and here he is looking a little bit sad in his armchair because Ramon wanted to be an artist, but his parents wouldn't let him be an artist. They actually wanted him to be a scientist. So he couldn't do art, so he was forced into science, and around the same time he got a hold of a microscope, which he's sitting with on his desk here. And what he managed to do was actually merge his interest in art with the science that he was doing. So he started to stain the tissue, animal tissue, with a silver stain, and was able to create these sorts of spreads of staining specifically the nervous tissue within animal tissue. And what he would do, this is before cameras were available, what he would do is draw what he saw in the microscope. So in these pictures, which are called Cajal diagrams, what he is drawing is the stained nervous tissue from different animal samples. These come from dogs, from cows, etc., whatever he could get his hand on. And he would draw what he saw, and this is actually revolutionary in terms of biology, because what it first provided an insight into is the fact that the cells within the nervous system are connected to each other in a very complex way. So we could say they knew neurons existed, they knew about the existence of the nervous tissue. It wasn't known before these diagrams arose that it was possible that they could be configured in these very complex arrangements. And these are also known as wiring diagrams, and what we can see is very complex connections between the cells. So here in this top left, we have all these cells lined up one against the next, and they're all connected through these very, very long projections. Yes, so I just thought I'd mention the backstory behind mapping cellular connectivity, both for the understanding of where this is coming from, but also the idea that you can have a diverse interest set and merge that with other aspects of your life, and you never know where things will take you as long as you remain interested and engaged, and I'll stop with that now. That happened around the turn of the century. Now, more recently, Sidney Brenner's lab did work mapping the entire nervous system of the C. elegans worm. And what they did was to take, this is the worm here, to take serial sections using transmission electron microscopy, and they created what's known as a wiring diagram, a complete wiring diagram of the entire nervous system of the worm. So here it is, this is the worm, and then they created here at the bottom, this is the network describing the connectivity between all the neurons in the worm. And what was really remarkable about this work was that they took this network and were able to perform topological analyses, which I'll describe to you in a moment. And from the analysis of the network of how the cells were connected to one another, they were able to predict the function of individual neurons. So what they were able to do is take the system, look at how the system was wired, look at how it was mapped out, and they predicted. Specifically, they found a neuron responsible for the worm's ability to smell, and another neuron for the ability of the worm to move backwards. And they were able to predict the function of these neurons based on their position within the overall nervous system. So this is the relationship between the structure of the nervous system and the function of the nervous system. And the fact that you can take a network-based approach to looking at how cells are connected to one another in a nervous system, or in a multicellular context, and predict the function of individual cells based on their positions. All right, so if you don't like maths, apologies, we're going to talk a little bit about maths here. So these are the network-based analyses we can do. So there are different levels at which these things can be looked at, either locally or globally. So the most simple local measure of a network one can do is to look at the degree of a node. And what this tells you is the number of connections any node has within a network. So it's the number of neighbors you have. So if you were to use like a Facebook analogy, this is how many friends you have on Facebook, and that's your degree. So what this tells you is how many people you can directly communicate with over that social network, or in the case of a plant tissue, how many cells one cell can communicate with within the tissue, because you can only communicate with your immediate neighbors. So this is what's known as a local measure, and it's telling you how important that cell is within that specific location. But there are other measures that we can look at to look at the higher order or more global properties of the network. And these include, these are some new words hopefully for you guys, something called betweenness centrality and random walk centrality. And the idea of betweenness and random walk centrality is it tells you, if you want to go from one point to another point, the best way to go, the most efficient way to go, is following the shortest path. So if you want to travel from point A to point B, you're going to follow the shortest path. And if you use Google Maps, it effectively routes you along the shortest path. So what betweenness and random walk centrality tell you are the roads or the nodes that you will traverse to go from point A to point B following a shortest path. So it's telling you which roads are positioned optimally within a network that you will want to traverse most frequently in order to go from point A to point B. And I'll just illustrate this for you here. So on the left, this multicolored psychedelic looking thing, these are some 3D generated computer cells. And then we can turn this virtual digital tissue that we've created on the left, we can turn this into a network where the nodes here, these are the nodes representing the tissue, the network representing the tissue on the left. So if we want to look at the degree, here we're false coloring the degree onto this virtual tissue. We can see these cells in the middle that are dark red, they have the highest number of neighbors, and the ones on the outside, which are in the blue, have the lowest number of neighbors. But say we want to look at this cell here in the bottom left. If we want to look at the degree of it, it has 5 neighbors, so we'll say that cell has a degree of 5, and that's how that calculation was performed. Now, if we want to look at betweenness centrality, what we're looking at now is the number of shortest paths a node lies upon. So let's have a look at that. If we want to go from this node to that node, the shortest path is following these nodes here. So because we are going from here to here, these 3 nodes get a score of 1 because they're lying upon the shortest path between these 2 other cells. And then if we go from here to here, this is the shortest path. Now this node now, because it's got a score of 2 because it lay on the shortest path between these 2 and now between these 2. So what you need in order to calculate betweenness centrality is you need a map of the tissue. You need to know where all the nodes are. And then with that knowledge, you can then calculate what the shortest paths are between each pair of nodes, and you do that for all of the nodes, and then you count it up, and you see how many times the nodes lie upon shortest paths between other nodes. Now, a variation of this is random walk centrality, and what this does is when you don't have a map, when you don't know what the topology of the network is, say you want to go from A to B, you can't say what the shortest path is because you don't have the map. But what you can do is send out 100,000 random walkers, so little Brownian motion walkers which will just go in random places, and look to see which ones took the least amount of time to go from point A to point B. And instead of getting a high confidence line to say this is the shortest path, you get kind of a fuzzy line to say, oh well, the walkers which followed these paths took the least amount of time. So you get something analogous to diffusion. If you were to diffuse molecules across a tissue, these would be the fastest way they would go from A to B. And this is important because in the case of biology, we don't have maps of tissues. For example, in a plant organ, one cell is not aware of where other cells are located because there are no maps present. So this is something more analogous to a diffusive process of molecules across tissues. Right, so these are higher order system level properties as opposed to degree, which is a local property. So these betweenness and random walk centralities, these are controlling the global flow of information across tissue. So if you want information to go from one bit of the tissue to the other, they would follow shortest paths, and these high centrality nodes of betweenness and random walk would be controlling the movement of these molecules. And if you'd like to read more about this, this is a paper we published some years ago in Interface. So given that they were able to identify the function of individual neurons in C. elegans worms, that's only ever been done in the nervous system, we asked the question whether you can do this in a plant. So can we uncover structure-function relationships in plant organs using a connectionist approach? By mapping the connectivity of cells in a tissue, can we predict the function of individual cells within a tissue? So to address this, we looked at the plant hypocotyl, which is the embryonic stem. And this serves two functions. One is to support the plant, and this is it right down here. So it's supporting the above ground part of the plant, and it also is involved in transport because it's the conduit between the above ground part of the plant and the below ground part of the plant. So you have water coming up here and say photosynthate going down there. So two functions here. If we look at the anatomy of the hypocotyl, it's not unlike the root. I'm actually showing you a root here, but the hypocotyl is effectively the same. And we're just going to focus on the epidermis here. So we have different cell layers. We have the epidermis, and then the cortex, the endodermis, and then the vasculature. In the epidermis, we have two types of epidermal cells. We have trichoblast, apologies for that, we have trichoblast cells, which are colored in green, and these cells make hairs. And then we have atrichoblast cells, and these cells do not make hairs. So it's there's one specific cell type that makes root hairs, which are involved in the uptake of nutrients from the soil, and we have another cell type which never will make a root hair. And what's interesting about this as well is that if you look at the cells which are making hairs, they're lying upon two underlying cortical cells, whereas the cells which do not make hairs are sitting on top of a cortical cell. So you can see the green cells are tucked in between two of these. What are those? I don't know what color that is. It's sort of flesh-toned cell cortex. So you have these green hair cells, and then the non-hair cells are sitting on top of them. So that's important, and I'll come back to that in a moment. So it's very well known that there are two cell types, and we know the genes that are controlling the creation of each of these hair and non-hair cells. So we know about how the pattern is constructed. But what we know less about is why this pattern exists in the first place. So what is the functional significance of this cellular arrangement? Why are there two cell types, and why are they located in the positions that they are? So what we did was take the Arabidopsis hypocotyl and turn it into the networks that I described, and we annotated the two different cell types shown in dark green and light green. The dark green ones are the hair cells, and the light green ones are the non-hair producing cells. And then we did this topological analysis. So the first analysis we did was to look at the degree, which is how many neighbors each cell type has. So we plot this here on the x-axis. These are bins of how many cells have between 3 and 6, 6 and 9 neighbors, etc. And then the frequency is on the y-axis. How many of them have that many number of neighbors? So what we can see is the non-hair cells, the cells which don't make hairs, have a lower number of neighbors than the cells which do make hairs, the trichoblasts. You can see the distribution here is a bit right shifted for the dark green hair producing cells compared to the non-hair producing cells. And the likely explanation for that is because the cells which make hairs, as I mentioned before, are sitting on top of two underlying cortical cells. So the cells that make hairs have more neighbors than the cells which don't. Then if we look at the betweenist centrality of these very same cells, what we find is that the cells which don't make hairs lie upon fewer shorter paths than the cells which do make hairs. So this is a little bit complicated, so I'm going to explain it one more time. What it means here is that because the non-hair cells, I said that the wrong way around, because the non-hair cells have a higher betweenness centrality, what that means is the non-hair cells lie upon more shortest paths than the hair-producing cells. So what that means is if you want to go from A to B and you want to follow the shortest path, that the non-hair cells are a better option than the hair cells. And this is completely opposite to the fact that the hair cells have more neighbors than the non-hair cells. Despite the fact that they have more neighbors, the non-hair cells lie upon more shortest paths. So you can ask, okay, that's nice calculation. What's the point of that? Well, we looked at the functional relevance of this by performing molecular transport assays. So we did this by taking seedlings and placing them onto media containing the fluorescent molecule called fluorescein, which is a molecule we're able to visualize in the confocal microscope, and then imaged it using confocal microscopy. And this is a cross-section of what we saw. So in white is the stain of the cell wall, and in red, these are the fluorescein molecules being transported. And what we can see is that the non-hair cells, which are labeled with A, have more of the fluorescein inside of them than the hair cells, which are labeled T. So these hair cells, which have more neighbors, have less of the molecule being transported from the root up to the shoot than the non-hair cells. And the non-hair cells are lying upon shorter paths than the hair cells. So this is confirming our prediction that these actually represent optimized routes of transport of molecules from the below ground to the above ground part of the plant. So what we've been looking at here then are higher order, you can say, emergent properties of epidermal patterning. So we have this pattern, and if we look at it and look at the next level up, how does this pattern create a function, a higher order function for the organ? This is what we've been looking at in these pieces of data that I've provided for you. So what we have is a topological proxy predicting molecular movement through complex organs at single cell resolution. So here the topological proxy is betweenness centrality, and it's predicting molecular movement, which is the fluorescein transport assay that I just showed you. And then this organ is a complex organ. And we can do that at single cell resolution. So by taking the network and analyzing the network using betweenness centrality, we can predict which cells will have more transport of molecules through them than other ones. So what we have then is an optimization in cellular patterning for the longitudinal molecular movement. So the optimization, if you think of moving from point A to point B, the optimal way to do that is following the shortest path. It's your fastest route. So the optimization in this sense is that some paths are shorter than others for the movement of molecules through the plant organ, and that's the optimization. So coming around to bridging structure and function, we've looked here at a higher order property of multicellular patterning in the plant hypocotyl. And what we're proposing here is that the fact that we have these two different cell types and they're organized in the way that they are, this represents a division of labor. So we have the cells which produce hairs, these green ones. These are taking up solutes and water from the soil. So their function is principally in uptake, but then they are juxtaposed, sat beside these non-hair cells, which never produce hairs, but they're optimized for transport. So the idea then is that these cells are receiving and taking up nutrients and water from the soil, and then shunting them over to these non-hair cells, which are specialized for the long-distance transport along the length of the hypocotyl. So if you'd like to read more about it, this is the paper, which is from 2017, a bit of time back now. And so in conclusion, what we have is a data-driven approach to quantify organ design and function in plants, and that this connectionist approach can predict molecular movement at single-cell resolution in plant organs, and that it has the potential to uncover higher-order structure-function relationships in cellular patterning in plants. And I'd like to thank the people who did the work, Matt Jackson, Salvador Andrade, Hao Shu, and Petra Stam, and the funding bodies who paid for this work, who include the Leverhulme Trust, the BBSRC, and the EU. And thank you all very much for listening to this presentation.